Yes. So today we are going to start our main central dogma process, and its name is the transcription and the translation. Okay. So if I talk about the transcription, so what happens in the transcription? So first of all, you know it very well. If you have a DNA like this, so if you have a DNA, so from the DNA you want to make a single stranded RNA, am I right?
Mm-hmm.
So this is the DNA and you want to make RNA, mRNA. So just tell me both these strands are useful for making this mRNA, like this mRNA is coming by using the both this strand or only the one strand of DNA?
Both.
Uh no, haven't I told you? Not both.
Oh wait, okay, then one.
Yeah, only one strand is using coming out. See. If
Oh, and okay, yeah, okay,
this
that's what
is
I meant.
DNA,
Okay.
so it is using only one strand of that. Okay, like this. And RNA polymerase, it is making something like this. So,
Mm-hmm.
so now this is mRNA. So only one strand is helpful for making the mRNA. Okay. So sometimes this question is also asked like suppose if both these strand help in the formation of mRNA, then you will have how many mRNA? Two
Of
mRNA, and these two
two,
mRNA are complementary to each other, and then they will
they
form the
make one.
double-stranded RNA.
Yeah.
Okay, and then they are sticking together, and if they are
Mm-hmm.
sticking together, then they are not going to form the protein. Okay, so when you have mRNA, so they will help in the formation of a protein chain, like chain of amino acid.
Right.
Amino acid chain is called protein. Got it? So this
Mm-hmm.
chain is called the polypeptide. Why it is polypeptide? Because it is they are joined by the peptide bond. So polypeptide chain. Okay, so this is a protein chain. Got it?
Yeah.
So from the MR, so if the yeah, if I make a schematic diagram, okay, so this is something like this. You have to remember that. Okay, first of all, suppose if I say if both the strand of DNA help in the formation of mRNA, then what would happen?
The tRNA would be not be there.
Then two strand will be formed.
Yeah, and then the
I mean,
tRNA would like not
two,
have the version.
yes, then you will get a two different type of mRNA and they are complementary to each other and they are going to stick together. And if they are
Double-stranded RNA.
yeah, and double-stranded RNA never forms the protein. Why? Because no tRNA will come, no mRNA will come. So that's why now no protein formation will take place. Okay.
Okay, so double-stranded RNA equals no protein.
Yes, because it should be open. One single strand RNA it is a open. So tRNA can come over like this. I told you, it is bringing the amino acid. So but if it is close, if it is close with that another mRNA, so now no tRNA will come over that. I will show you the video, don't worry. Okay, suppose this is the DNA. Okay, so I told you this is 3 cap to 5 prime, like 3 prime to 5 prime, 5 prime to 3 prime. So you should know one thing. There should be one promoter at this side. One promoter. Promoter means what? I told you.
The starting of the RNA
Yes.
transcription
Can we say
process.
like transcription is not going to start randomly? It is starting from some specific location, and this is called the promoter. And we
Hmm.
have a specific specificity here. This is called the terminator, right? So what is the meaning of this terminator? If I am making
It
like this.
ends the it ends the transcription process.
Yes. And in between them we have what?
The RNA.
Yeah, the structural gene, right? So
Yeah,
these
structural
are the
gene.
structural gene of DNA which help in the formation of RNA, right? So all
Yeah.
these will help in the formation of RNA. So can we make like this? Can I make like this? So from here to here, you will get the mRNA, right? So this is your
Oh yeah.
mRNA. So mRNA will get formed in the structural gene portion. Okay. So remember always, uh, you should know in the DNA, first strand is known as the strand, template strand, and the other is known as a coding strand. So one is template strand and the other is coding strand. Okay, you have to remember that template strand and the coding strand. Okay,
Mm-hmm.
So now you should know one thing. Of course, now mRNA is coming from that. mRNA is coming from the DNA. So you will call it as a mRNA is coming from the DNA. So this is known as a transcription, and you should know one thing. Which enzyme we need? RNA polymerase or the DNA polymerase?
RNA polymerase.
Yes. So here, and this is completely independent DNA dependent RNA polymerase. You will call them RNA. It is not depending on the DNA. So that's why it is a DNA independent. Oh, sorry. Actually, it is depending on the DNA. Okay, and then it is forming the RNA. Like
Well,
because of
yeah,
the DNA, RNA is
it
coming. Na, so
separates
so
from the
yes. So
DNA.
so you will call this enzyme DNA dependent. DNA
Mm-hmm.
dependent, because if there is no DNA, then it can't form the RNA. So that's what is known as a DNA dependent RNA polymerase. Okay.
Mm-hmm.
Got it. So can we say it help in the polymerization of the mRNA, which is coming from the DNA? Okay.
Yeah.
And in the DNA there are two strand, template strand, coding strand. So they are coming with the help of that. Okay.
Mm-hmm.
So okay. So now I'm going to show you some structure. Okay. Like how, like firstly we are talking about the prokaryotic, how transcription in prokaryotic take place. So see, if you have a DNA, we are talking about the prokaryotic organism. Okay, so this is your DNA, but I'm talking about prokaryotic, right? You are not prokaryotic. So this is the first, there is a initiation process. Actually, you know it very well. There is a promoter. There should be a
Mm-hmm.
promoter at this side, right? So promoter
Yeah.
is there. So now it want to start the process, but it is not being started. It is not getting started. I don't know why. So what should I do? Actually, even after using the promoter, the DNA is not getting started. So, do you know what is happening? So, you know it very well. There is an enzyme, DNA, like RNA polymerase here, right? There is a RNA polymerase. So, RNA polymerase help in the like they are going to come at the promoter site to start the process of mRNA, but still nothing is happening. So now there is a factor here, and this factor is known as a sigma factor. Remember, which factor?
Sigma.
Sigma factor. So this sigma factor is going to come in this RNA polymerase, and then when they are going to come here, then the process will start. Okay. So RNA polymerase is normally, RNA polymerase is always coming at the promoter site and starting the process, but it is not started. Why? Because we need something, and that something is known as what? We
Sigma
need something.
factor.
Yes. So there is a sigma factor which comes under the RNA polymerase, and when it is coming here, then the process being started.
What is a
Okay.
sigma factor?
Actually, it is something. Okay. I have to show you the video. Then you will see. Actually, it is something. Like there is some substance. Okay,
Uh-huh.
which should be present with the RNA polymerase. Without it, process will not start. So we have
It's
given
like
that
it's
something.
like it's like a key to a car.
Yeah,
Is
you
that
can
the
say that. It's like a
okay?
key when this going to attach with the RNA polymerase, and then it is going to start. Then the transcription is started. Okay. So now because of this sigma factor, it is like a something push. See, just tell me. Suppose if my car is not working, so sometime it happens like if you push the car and then it is going to start.
No.
Sometimes car becomes like the na, like it is not being started, but once you just push it the car, so suddenly it started.
I don't think that's how it works, but okay.
Sometimes it happens. Okay, suppose you have a bike. Okay, I don't know
Oh.
what is happening, like if you're doing something like, so it's not being started, but whenever you push it and key is on, key is on, and when you
Mm-hmm.
push it, so suddenly it's engine like start working quickly, something like
Mm-hmm.
that. So it was just a push. So because of push,
Mm-hmm.
your vehicle usually gets started. Okay. So
Okay.
just a push is it needed, push. So that push is called the sigma factor.
Do
Okay.
you have a motorcycle?
Now I have the bike.
Is
Motorcycle is
that?
bike, yeah, but yes, I have.
Oh, damn, that's cool.
But I'm not talking about myself, okay? I'm
Oh
talking
no, no,
about
no,
the
no, no.
okay. So this is the sigma factor, okay? And now the process being started here. See, see here. Now from this portion, new mRNA is coming. Am I right?
Mm-hmm.
Yes. So this mRNA is coming. So it is the initiation when the sigma factor is coming. So at somewhere, okay, because the now RNA is going to be started, RNA formation. So somewhere you will see there is a phase which is known as the elongation.
Mm-hmm.
I told you it needed a push. That's why sigma factor came, right? It needed a push. So when the car is already started, so do you again need a push?
No.
No, so it means now the process in the middle, okay, this middle process known as a elongation.
Sigma factor's gone.
Yeah, so sigma factor will remain inside or sigma factor remain outside now?
It's outside.
Yes, so now no push is needed. This factor
Mm-hmm.
is needed only at the beginning, right?
Mm-hmm.
So but RNA polymerase should be inside or outside? RNA polymerase.
Inside.
Yes, RNA polymerase should remain inside. Why? Because it help in the formation of what?
RNA or
Yes.
proteins.
So it would look something like this. Okay, if I talk about, so just give me 1 minute. So it would be something looking like this. Like here, it has reached, it is the mRNA. So if the mRNA has reached here, from here to here, mRNA is reaching here. So can we say like the new mRNA chain has formed from here to here? See. One second, one second. This is the, oh my God. One second, just.
Mm-hmm.
Yeah. So this is known as a RNA polymerase. RNA polymerase, I just wrote there. RNA polymerase. So if the RNA polymerase has reached here, from here to here, spotlight, if the mRNA, like the RNA polymerase started from here, right? RNA polymerase. Now it is coming here. Finally, it has reached there. So can we say a new chain has already formed mRNA from here to here?
Yeah.
So that's why now your mRNA chain is like this.
Mm-hmm.
This is mRNA chain. So this mRNA chain keeps on increasing or keeps on decreasing if it is going further.
See it
This
again.
this RNA polymerase is going further, right? From left to right.
Yes.
So the
Yes.
mRNA chain length is going to increase or going to decrease.
Grease.
Of course, it is going to increase. Why? Because it is help in the formation of mRNA, na? So if it is going ahead, so of course the length is also going to increase. Now, so this is the elongation phase. And
Okay.
just after the elongation phase, what should you see? So
The
now,
coming out, the unraveling phase.
yeah, so it's an, can we say that now that there is a termination phase? So
Oh
I'm,
yeah, that phase. The single strand comes out phase.
yeah. So now, it's just a rough diagram. Okay, now it is going to the termination. So actually there is like the process not going to be terminated. Okay, so again it needs some factor. Okay, again it needed some factor to stop that. So finally it has reached there. So mRNA length would be higher or smaller? Of course higher, from here to here. Can you just see that the mRNA chain has already formed? So this mRNA chain is coming from one strand or both the strand? I told you.
One strand.
Yes, one strand. Okay. So now here there is a RNA polymerase, and this RNA polymerase actually needed another factor, and this factor is known as a rho factor. Rho,
What?
rho factor, rho. Rho factor. Initially, you had a sigma factor, right?
Mm-hmm.
Which is starting, and there is another factor which is terminating, and this terminating factor is known as a rho factor. Rho. Okay?
Using all these fancy Greek symbols.
Yeah. Okay. So if I ask you, if you have a sigma factor, so sigma factor help in the what? Which pro?
Initiation phase.
Yes, initiation of the transcription, right? And if
Yep.
I talk about the row factor is helping in what?
The termination of the, yeah.
Yeah, okay. So we have these two factors, and you should remember that. So the RNA polymerase reaches the terminator, and the RNA polymerase falls off there. Okay, but remember this termination is facilitated by some terminating factor, and this terminating factor is known as a Rho factor. Okay, so in the
Yeah.
prokaryotic mRNA synthesized, so it doesn't require any further processing. Okay, it is quite active, and it is fine now. So this is the good mRNA, but in our body, eukaryotic, like this mRNA seems like quite good. Okay, this is completely fine and it is ready for the translation process. But now in our body, we are not getting mRNA actually. We are getting quite complicated mRNA. It needs some processing. Okay,
Hmm.
so these things are happening. Okay, this which is really good. So first of all, I want to tell you something. Just give me 1 minute. Just one
Before you
minute.
can drive on the roads, you have to get your license first. Just like, is that just like kind of like that?
You can say that.
Not
Okay.
really. I don't know.
Okay. So actually, okay, you are asking to me.
Oh, no, 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 no. I'm just
Okay.
saying, like, is
Okay,
that like
okay.
an equivalent analogy
Yes, yes.
to this
Okay.
situation?
Okay. I thought you were saying, so you whenever you go to the roads, do you need license? Of
Oh,
course.
yeah.
Okay, so yes, here these are the two factors which we have just studied that. Okay, so of course RNA polymerase is also needed, and now I want to show you a very good video. 1 second, yes. So this is very much needed. So same thing is happening in our body also, but just tell me, do we get the uh, very functional mRNA or very complicated mRNA in eukaryotic organism?
Uh,
Very
Complicated,
complicated one.
sophisticated.
Yeah, so you will say it is a hnRNA. hnRNA you will say. Okay. So hnRNA means a heteronuclear RNA. So
Hmm.
heteronuclear RNA means which is very complicated. Okay, so I will tell you, don't worry. So before that, just see this once. Can you just see that?
Yep.
Strand of DNA to mRNA is termed as transcription. In transcription, only a segment of DNA and only one of the strands is copied into RNA. Both the strands are not copied. If both strands act as template, they would code for RNA molecule with different sequences of the code for proteins. The
See, I told you, Vishnu, like in the DNA, one strand is known as a template DNA, and other is coding strand. I told
Mm-hmm.
you. So this is the template DNA. Template DNA help in the formation of mRNA. Okay.
Yeah.
Template means like whenever you go for the photocopying, so you should give one paper, and from one paper, so many papers are coming. So that one paper is called the template, right? So from the
Mm-hmm.
template DNA fragment, RNA is coming. So both these 10 can work as a template? No, we need only one fragment to be template, one fragment to work as a template, right? Because we don't
Mm-hmm.
want to make double stranded RNA.
Right.
sequences of amino acids in the proteins would be different. Hence, one segment of DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. The two RNA molecules if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into proteins, and transcription process would become a fertile one.
Hmm?
Transcription unit. The
Yes,
transcription
yes.
unit.
You are saying something?
What does it mean by fertile one?
Which one?
It said, yeah, this would prevent RNA from
This
being
would
translated
prevent
into.
RNA being translated into the protein, and transcription process would become a futile one actually. I mean, you can say that which is not useful.
Okay.
Futile. Okay,
Okay.
like it is not, because if there is a two-stranded RNA, so it means that they are forming double-stranded RNA, and
Mm-hmm.
double-stranded RNA can form the protein. No.
Oh no.
Why? Because it won't allow the tRNA to attach on that. And if it is
Yeah.
not allowing tRNA or rRNA, then there is no formation of protein. So that's why then no transcription process would be there. I mean, you can say transcription is already done, but translation will not happen. Transcription is good, but translation will not happen. Okay.
Yes.
So we need double-stranded RNA or the single-stranded RNA.
Single.
The transcription unit of
I
DNA
already told you
has
these
three
things.
regions. 1.
Yeah.
Promoter. 2. Structural. 3. Terminator. The double helix DNA contains certain specific genes called promoter sites at which DNA unwinds due to temporary breakage of hydrogen bonds. The promoter is located towards 5' end upstream of the strangular gene. The two strands of DNA have opposite polarity, and the DNA-dependent RNA polymerase
I
catalyze.
told you, right? DNA dependent polymerase. You remember that?
Yep.
Why it is called DNA dependent polymerase? Why?
Because what? What did you
Why
say?
why this is
Because
called?
RNA is dependent on the DNA.
Yes, because this mRNA is coming from the DNA, na? So if it is
Yeah.
coming from DNA, so of course it should be DNA dependent. Okay.
Oh yeah. I see. Yep.
Yes. So yes.
The polymerization in only one direction. Transcription is also called DNA-dependent RNA synthesis.
This red is known as a RNA, right? So just tell
Mm-hmm.
me, it is basically attached both of them or only one strand of DNA? See, very carefully. It is attaching to both of them or only one thing? See, this is the strand of DNA, right? And it is coming from there. It is not attached
Oh,
with another one.
it's
Can you
just
just see
one of
that?
them.
Yeah, one of them. Green is not attaching on that. Okay,
Mm-hmm.
there is a gap. So it means it is
Yep.
coming out from the one strand only. And this, so that's why this strand is known as a template strand. Okay,
Okay.
because from this mRNA is coming.
And the enzyme RNA polymerase is also called transcriptase.
So
This
RNA polymerase help in the formation of RNA, right? So that's why it is known as a transcriptase, because the process name is transcription. Okay,
Mm.
transcriptase. So Vishnu, is this clear to you?
Yeah, but I'm gonna have to rewatch like the recording again,
Okay, okay. So
just for revision.
okay, and can we say that, so how, which factor I need?
Say it
Factors
again.
at the starting,
prime AT
which factor I need? There are two factors, na, I told you, beginning at the ending.
Oh, it's I got it. Sigmoid and rho.
Yeah, sigma factor at the
Oh,
beginning
sigma,
and
not
rho factor
sigmoid.
at the terminating, right?
Sigma and rho.
ATGC, ATGC, ATGC,
These are the
ATGC,
codes. These are the
ATGC,
codes.
ATGC to 5' coding
They're
strand,
random, right?
5' TSCG, 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 3' promoter in a transcription unit defines template and coding strands. By switching its position with terminator, the definition of coding and template strands could be reversed. The terminator is located towards.
Yeah, so it is done now. Okay, so this is basically about the prokaryotic, right?
Yeah.
Prokaryotic. But if I talk about not only the prokaryotic but also the eukaryotic, okay, so same thing is happening here. But I already told you, whenever we get the mRNA, so our mRNA is not so easy. Our mRNA is quite complicated, right? So if I talk about our RNA, so this need something, so I want to show you here. See, whenever we get the mRNA by the same process, so this mRNA actually contain two things.
Mm-hmm.
First, it contain the
And then
introns, and
extrons,
contain exons.
extrons.
You have you heard that?
Yeah. And the first
Okay,
one. The first one.
first one means, why?
The
I
first
mean,
video
where you have studied that?
you video it said intron and extron.
I mean, have you studied these things in the school?
No. The first video you showed me,
Okay, 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 okay,
transcription, that
okay,
one.
okay, 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 okay. I got it. I got it.
Yeah. Remember?
Okay, I thought you have
The
already
orange
studied.
and yellow?
Yeah,
Orange
yeah,
and
yeah,
yellow?
yeah, yeah, yeah. Got it.
Yep.
Okay, so in the mRNA, which we are talking about for the human being or the plant or the eukaryotic organism, so we have the two strand, introns, exons. Okay, so actually, here exons are very good. Exons are very much needed. Exons are very good in terms of the yes, like if the exon is present there only, so that is a real mRNA which is needed for the human being. Right now it contain like, and the introns are very bad, and we don't need
Mm-hmm.
introns at all. We are not liking introns, so we want to remove these introns. So it means, if I
Mm-hmm.
talk about in our body, okay, so our body, the mRNA is going to form, but it contains both these things, introns and exons. So that's why
Mm-hmm.
you can't say it is a complete mRNA, you will call them hnRNA. hnRNA means heteronuclear RNA. Why? Because it is containing only good things or bad thing also,
Bad both.
both. So that's why you will not say this is mRNA, you will say this is a heteronuclear RNA. So we have to remove what? Introns or the exons?
Introns.
Yes, so we have to remove the introns. So that's why so how can we remove them? So we can remove them by making them like a ring. You don't have to cut them, never ever cut them. They should remain fixed, and then there is a cutting process. Like what? Like it is, it will something look like this. See, just give me 1 minute. Exons are basically red color, like this.
Mm-hmm.
And we have just making a ring like structure for the introns. See, like this. Are you getting? We have, like, if you have a okay, I'm going to show you like this. See this, can you just see that?
Mm-hmm.
See, if we want to remove that portion, so can we make like this, like this?
Mm-hmm.
So now good portion are attaching, and this is bad portion, so now can we cut them?
No, there's no electron. You can't cut them off.
See,
Because
there
if
is a
you cut it
line.
off, then the electricity won't flow to your phone.
No, this is the thing.
Wait. Show me your cable again.
Now can you see that? Can you see that now? Now this thing,
Yeah.
we want to remove this sky blue color. So if you,
Okay.
so you can't cut directly, you have
Yeah.
to cut when red portions are attached. So now just attach this and this.
Okay. Yeah.
Okay,
Maybe the cable wasn't the best example because
okay, fine. So now
yeah.
it has formed, and now you can cut them, yes or no, the bad one,
Yeah.
because the good and good one are connected. So
Mm-hmm.
that's why the good one are connected, and now they are forming a ring like
Remove
structure like this, right?
introns.
Now we are
The
going to cut
one.
them. So this process known
The
as a RNA splicing.
oh, splicing. Okay.
So in the RNA splicing, we are going to remove what? Introns or the exons?
Introns.
Yes, because introns are non-coding thing.
Shitty. I
It
mean,
means
bad.
introns means they are not helping in the formation of protein. So that's why these introns are need to be removed. So we are removing them. So this is known as the introns. And you are going to get only the red one because these are removed now. So if these are removed now, so we are going now we do so see some processing has been done. We have just make the RNA which contain only the introns or the exons. Only
Hmm?
exons, right? Only exons is contained, na? This hnRNA. Only
Yeah.
exons are because introns are removed. So we need to add some cap or tail for the stability. Okay, so you have to add something at the top here. So this process known as a capping. So in the capping, we add a molecule which is called the sorry, which is called the methyl guanosine triphosphate. Just say it, methyl
Methyl guanosine
guanosine triphosphate.
triphosphate.
So whenever
MGP.
yeah, so methyl guanosine triphosphate,
Triphosphate.
actually whenever there is some changes is going to happen in there, so we are just providing these are the stability factors, you can say, methyl guanosine triphosphate.
Where does GEP? Oh no.
Methyl guanosine
GEP.
triphosphate, just give me 1 second, these are the capping.
Okay.
And at the end we are adding some tail, and at the tail you will call it as a poly tail. This poly tail means adenylate residues. I know you are thinking, what is that? Don't worry, I will show you something. Poly a tail, okay, and this is known as a adenylate residues. Adenylate residues. So this process is known as a tailing. So is this clear to
Hmm.
you? Like we are adding the why we are adding these, because we have just removed the structure, and we have just removed the structure. So there are these factors are there. Uh, like what? We have just used the process after this placing capping and tailing. So in the capping and tailing, you know it very well. In the capping, which thing we are adding actually? Methyl guanosine
Guanosine
triphosphate.
triphosphate.
And at the tailing, with what which thing we are adding?
Uh, adenine.
Adenylate residues. Okay.
Yeah.
Okay.
So
So
capping. Let me just write that down. Capping. Capping. You're adding methyl guanine MGT
hmm,
and GTP.
hmm.
Capping. Capping. Methyl guanosine. Methyl
Guanosine triphosphate.
guanosine tri.
Hmm.
Why is everything in like P3 or like 3P?
3p?
Like triphosphate. So like is that an oxygen too?
No. It's nothing like that.
P1 or 3P? I don't know. Okay, triphosphate. And then poly or
Tail.
tailing
Polyetail is known as a adenylate residues. Okay.
adenyl
Adenylate
adenyl.
residues.
Okay.
So yes. So this is how I'm going to show you something. See, एक second.
I've been fascinated
Just
by how prevalent this bullshit idea is around the concept that to be successful in life, it's about your education and
1 second.
Is that an ad?
1 second.
Alright.
Can you see that?
Yeah.
and the splicing process. So in this picture, we're looking inside the nucleus. This is going to be our DNA, and this is the nuclear membrane. The first process is transcription of DNA to make hnRNA. We're going to have to add three main modifications to the hnRNA to make an mRNA. The first of which is capping using methylguanosine.
Yes, Vishnu, you have just seen that like in our body, in human being, hnRNA is forming the mRNA, right? After doing the capping, splicing, and tailing.
Hmm.
In human being, do we directly get the mRNA? No.
No.
The further processings are needed, right? Which are the splicing, capping, tailing.
Yes.
Okay. So
Yep.
this is saying the same thing.
This
Capping.
process mutes the mRNA and makes it
Can you say
only usable
cap is
when the
there?
cap is removed.
Hmm.
And then the splicing process follows, where we remove the introns using snRNP or small ribonucleoproteins. Keep in mind antibodies to snRNP are called anti-Smith antibodies, and they're highly specific for SLA. And finally, tailing by adding the nucleotide sequence AAU-AAA. The tail allows the mRNA to leave the nucleus. So once we have an hnRNA that's been capped, spliced, and tailed, we call this an mRNA. This fancy little molecule leaves the nucleus to be stored in P bodies in the cytoplasm.
Yes. So now these are the further explanation we will tell you. So first of all, you should know Vishnu, like in our, if I talk about the eukaryotic organism, so our RNA basically contain hnRNA. hnRNA means
Hmm.
hydronuclear RNA. So it contain introns and exon both of them. So we have to remove the introns because these are the non-coding things. They don't help you in the protein formation. So that's why we have
Yes.
to remove them. So we can remove them like this. This is known as the splicing. So this is the basically at the middle, but in the beginning and the ending we have to do something else. What? So in the beginning we have to take the capping, and
Mm-hmm.
at the last side we have to do the tailing, right? So in the tailing we have to add the adeninated residues, and at the end we have to add the methylguanosine triphosphate, right? And this is how
Mm-hmm.
you are going to get finally the mRNA. Okay, so which is
Yep.
complicated, the I mean the,
HNRNA.
yeah, and in the, so which process typical, prokaryotic or the eukaryotic one?
Eukaryotic.
Yeah, because in the prokaryotic it was very simple. They directly get the mRNA, right? So we can say
Mm-hmm.
the hnRNA undergoes two additional processes.
Yep.
hnRNA, capping and tailing, and before capping and tailing, there is a further thing is placing. And
Mm-hmm.
remember, sometimes the question is asked how many arylated residues we are adding. So you have to remember 200 to 300. These are the residues, adenylate residues. Okay, remember that. So finally, this is completely, so finally we have fully processed the conversion of hnRNA to mRNA. So from
Yep.
hnRNA, we are going to get mRNA here. Okay, and now finally it is released from the nucleus into the cytoplasm. So now this is coming into the cytoplasm from the nucleus. So from the nucleus, now it is coming to the cytoplasm. Got it now?
Yep.
Okay, very good. So now, okay, if I tell you something, of course, we have the DNA or we have the RNA. So, of course, they have some code on that. Like you also have some code, and your code is similar like your parent, like AT, something like this. Maybe
Mm-hmm.
my code is 80, something like this. So maybe I have, of course, I have some different code because my code is look like my parent. Your code look like your dad and mom, right? So this is known as a genetic code. So I'm going to discuss about that. See, so suppose if you are getting the mRNA and mRNA code is like this. Oh, in the mRNA there is no T, there is U, right? I told you mRNA doesn't contain thymine, it contains uracil. DNA contain adenine, DNA, adenine, guanine,
ATGC.
yeah, ATGC. But
Some has
remember, but
AUGC.
in the, yes, so it contain the AUGC. There is no thymine in the mRNA. Okay,
Why?
why? It is property. It is the
Why?
property of that. thymine is not there. For the DNA, it has this property, like they contain only the four type of these. So it is the property of DNA. In the property, I like this pen. Why I like this pen? I don't know. It is my property. So property of DNA, it contain the thymine. I mean, it contain the oh, here I just make the thymine. So DNA have the property of containing these two nitrogenous bases, and mRNA contain these four nitrogenous bases, not the thymine. So it is the property, and that's why you can differentiate between them. Then if they are same, so you won't be able to differentiate which one is mRNA, which one is DNA. So that is the unique property by which you can differentiate them. Okay. So DNA contain
Mm-hmm.
thymine, but mRNA contain uracil. So there is a code, and this code is coming from the DNA. Am I right?
Yes.
Okay. So now, you should know one thing. You should know one thing. This is the complete genetic code. Okay. So because of that, this genetic code, of course, there is a formation of specific type of protein in our body. There is a
Yes.
formation of specific type of a protein, like there is a code is going on. So it means our DNA has some code, and this code help in the formation of a specific kind of mRNA also. Yes or no?
Yeah.
So this genetic code, we will discuss this thing in the next class. Okay. So right now you should know our DNA has some code which is similar like parent. Why your protein look like your mom and dad? Because you have this code, and this code is
Similar
coming
code.
from, yes, similar something kind of code from mom and dad. That's why you have a similar kind of protein in your body. Okay.
Mm.
If you don't have that code, then you are not going to show some similar character. You will show the character
I
of other
see.
maybe. Okay. Now,
Yes, I understand, sir.
okay, so we are just ending it here. Okay, rest of the portion we will discuss
Mm-hmm.
in our next class. Okay.